Fortnite gods, have mercy on me.
3, 2, 1, drop it.
Hello, my name is Sean Gao,
I'm Christopher Zhu.
and today we're going to be reviewing molecular genetics. So first we're going to go over the cell cycle, which consists of mitosis, meiosis, and DNA replication. And here's the cell cycle, and as you can see, the largest part of the cell cycle is interphase, and the smallest part of the cell cycle is the mitotic phase. And so starting at the G1 phase, DNA is synthesized, organelles are duplicated in the cell, and the cell grows in size and creates building blocks. Now moving to the S phase, DNA is replicated and centrosomes are duplicated. After that, in the G2 phase, the cell makes more proteins and organelles, and this phase ultimately prepares the cell for mitosis. And at the end of interphase, chromosomes are uncondensed. So here's the stages of mitosis, and in the beginning right here, in prophase, chromosomes are tightly coiled, and sister chromatids join together, and the mitotic spindle fibers begin to form. Next is the prometaphase, and in this phase, the nuclear envelope opens, allowing microtubules from spindle fibers to interact with the condensed chromosomes, and kinetochore, a protein structure located at the centromere, receives kinetochore microtubules from each pole. Following prometaphase is metaphase. And in metaphase, the spindle fibers push the sister chromatids to the middle and arranges them along the metaphase plate. After metaphase is anaphase. In this phase, centromeres divide and each chromatid is pulled apart by spindle fibers, separating the chromatids. And these chromatids move towards the poles of the cell. After that, in telophase, chromosomes are at the complete opposite ends of the cell, and new nuclei are forming on each side of the cell. And there are 46 chromosomes within each cell. And for an animal cell, a cleavage furrow forms, as you can see in this picture. But in plant cells, a cell plate forms. And in cytokinesis, the cytoplasm divides, ultimately splitting the cells into two. Next, I'll be going into the process of meiosis. So here in prophase 1, each chromosome pairs with its homologue, and crossing over occurs at the chiasmata, and microtubules attach to the kinetochores. In metaphase 1, the pairs of homologous chromosomes line up in the middle at the metaphase plate. Following metaphase 1 and anaphase 1, connections between homologs are broken, and the homologs move towards the poles, guided by the spindle apparatus. And in telophase 1, each cell is a haploid, and following the telophase, in cytokinesis, the two cells are completely separated due to the division of the cytoplasm. And the next stage of meiosis is prophase 2. And in prophase 2, the microtubules attach to the kinetochores of the sister chromatids, and they move towards the center. In metaphase 2, the chromosomes are positioned along the metaphase plate. And in anaphase 2, cohesions hold the sister chromatids together and are broken, which allows the chromatids to move towards the opposite ends of the poles. And as you can see, the sister chromatids are moving away from each other. And in telophase 2, the parent cell results in 4 haploid daughter cells, which are distinct from the parent cell. And in cytokinesis, the cytoplasm will divide, which completely splits the cytoplasm, ultimately forming 4 unique cells. And so some differences between mitosis and meiosis is that mitosis only has 1 division, while meiosis has 2. As previously stated, mitosis has only 1 stage, while meiosis has 2 stages. And crossing over only occurs in meiosis during prophase 1. in mitosis results in 2 genetically identical daughter cells, while in meiosis results in 4 genetically distinct daughter cells. And meiosis contributes to genetic diversity through independent assortment, random fertilization, and crossing over. But in mitosis, there are identical somatic cells for growth and repairing damage. And in meiosis creates sperm and egg cells, which are gametes.
Now we will talk about the process of transcription, which is the process where DNA is transcribed into mRNA. So transcription begins with the transcription initiation complex, where a sequence of DNA, usually with the sequence TA, attracts transcription factors and RNA polymerase, which facilitates the binding of a promoter. An RNA polymerase unwinds the DNA while elongating the complementary RNA transcript from 5' to 3'. In the termination phase, RNA polymerase codes a stop sequence in the gene, and this completes the mRNA strands, causing it to detach from the DNA. However, there are still post-transcriptional modifiers before the mRNA is exported to the cytosol. One of the post-transcriptional modifiers is the addition of a 5' cap to the 5' end of the mRNA, and this alerts ribosomes to begin translation. The 3' poly A tail protects free mRNA from degradation and signals the export of cellular functions the transcript needs from the cytoplasm. The other post-transcriptional modification is alternative RNA splicing, in which introns are removed by spliceosomes, allowing exons to splice. This results in different proteins being made due to the different combination of exons and introns. The last thing we'll talk about is translation. In translation, after the mRNA is modified and exported, it is exported to the cytosol, where a small and large ribosomal subunit attached to the mRNA which initiates translation. An initiator tRNA first binds to the P site of the large ribosomal subunit, and other tRNAs bring corresponding codons into the A site. Next, the growing polypeptide at the P site is then transferred to the A site, and the tRNA that is still at the P site is moved to the E site where it is released, while the tRNA at the A site moves to the P site, continuing the process until a stop codon terminates translation. In bacteria, transcription and translation can occur simultaneously since bacteria do not have a nucleus. However, post-transcriptional modifiers can occur in this situation.
Thank you guys for watching this video. Make sure to subscribe and annihilate the like button. And thank you, Mr. Rock, the absolute GOAT, for teaching us this year. And can I at least get like 2 likes? Like, I think just 2 likes, guys. Just 2 likes, please, please.